The overall goal of the following experiment is to image neural cells at high resolution in the zebrafish embryo. First, DNA-encoding membrane-targeted green fluorescent protein is injected into a one-cell stage zebrafish embryo, which results in mosaic inheritance, thereby enabling enhanced imaging of scattered cells. Embryos are then immunolabeled with anti-GFP and sectioned, enabling visualization of the morphology of single cells at high resolution in a fixed preparation. Alternatively, live embryos can be examined using time-lapse video microscopy in order to image cellular dynamics. Results are obtained that show cell morphology or dynamics. Here, the zebrafish hindbrain is imaged with confocal microscopy.
Hello everyone, I'm Pradeepa Jayachandran from the laboratory of Dr. Rachel Brewster in the Biological Sciences Department at the University of Maryland, Baltimore County. Today we'll show you a procedure to label and image neural cells in zebrafish embryo at high resolution. We typically use this procedure in our laboratory to study the mechanisms of neurulation in zebrafish embryo at a cellular level. So let's get started.
Place wet Kim wipes around the edge of a 100 mm petri dish. Carefully secure a glass microinjection needle on a strip of wax inside the humidified dish. Under a dissecting stereo microscope, align the needle against a graduated slide. To obtain a tip of the appropriate size, use a clean razor blade to gently tap the needle where it has a 1 micrometer diameter. Connect the needle to the microinjection apparatus.